the title of our session is New Life Sciences, Understanding the Essentials of Life. We are having two very exciting speakers, and our rapporteur is Dr. Zoe Kwan, who's the she's a founding board member of Catalyzing Collaboration, which brings industry and academia together and to try to find startup company opportunities and applications. And she's also president of King Chu Thinking, which combines strategy, international business, and assessing technologies for their growth potential in order to help companies determine whether or not they need, where they need to be, where they need to improve, and so forth, which is a big job. And so her particular interest is in innovative research, and we're very grateful that she is going to be our rapporteur. Thank you, Dr. Kwan. So as you all know, and as we heard from all the sessions that we have attended so far, that the human genome, of course, is proving to be a whole lot more complex than people had expected with all the variations and the epigenetics and so forth. So there has to be new approaches and very powerful computational means to be able to understand the complexity of the human genome, and then bioinformatics that helps translate and bridge it to what is the meaning of these complexities. So this is what this session theme is about, okay? We are very honored to have two speakers with us for this session. The first speaker, honored to introduce him, Dr. Omen Gilbert. This is Dr. Gilbert's fourth visit to the Bibliotheca Alexandrina. Omen. Okay, Gilbert. Oh, first. I'm dyslexic. Okay. Even though I know of you. Trust me. It's Dr. Gilbert Omen, and this is his fourth visit to the Bibliotheca Alexandrina. He is a professor of medicine, genetics, public health, and computational medicine and bioinformatics at the University of Michigan, Ann Arbor. He is former deputy science advisor to the U.S. President Jimmy Carter, one of my favorite presidents, and he's also former president of the American Association for the Advancement of Sciences and American ASSN, American Cancer Research, and director for the Center for Computational Medicine and Bioinformatics. His research, he has a long-standing interest in mechanisms of genetic predispositions to risks from environmental and occupational exposures, pharmacogenetics and pharmacogenomics, and science-based risk analyses. His research focuses on cancer proteomics. He is indeed the former president of the American ASSN Cancer Research, which I mentioned already, but he's also involved in clinical informatics, databases, and computing several major projects, medical and translational research, proteomics, systems modeling, systems biology, and he led several major projects, and especially in the proteomics area, and leading the Proteomics Alliance for Cancer Research, the Human Proteomic Organization, and Proteome Project. And I can go on, but I think you should hear from him, not me. Thank you so much. It's an honor to have you.
Okay, well, it's a great honor for me to be back here in Alexandria and be part of this BioVision 2012. Thank you all for coming. You can see here that my logos are for the University of Michigan, and for the Center for Computational Medicine and Bioinformatics. In the United States, we're required to give these disclosures. Suffice to say, I have no conflicts of interest in this lecture. This place, Bibliotheca Alexandrina, is a major international institution. Its symbolism, its architecture, its leadership, and its role here in Egypt and in the Muslim world and internationally is of great importance. So I commend you all, and it's a pleasure to be engaged in this activity with Ishmael and with all the rest of you. You're well aware that Dr. Sarah Galdin is a leading spokesman for the Muslim world and the need to recapture the spirit of science of 1,000 years ago. It's reflected in these comments from an important piece he had in Science magazine, our AAAS magazine, in 2008. The last line, science requires freedom to inquire, challenge, think, and envision the unimagined, is exactly right. And it matches this comment from Nelson Mandela, which we saw this morning, that education is the most powerful weapon which you can use to change the world. With regard to living things, there are two overriding concepts which were the basis of the textbook that was used at Princeton University when I was a first-year university student, and the course was taught by Colin Pittendrigg. That is, number one, the capture, storage, and utilization of energy, and number two, the reproduction of individuals and the reproduction and evolution of the species. These are very valuable ideas to organize the tremendous amount of information we lay on students and think about as we try to understand the complexities of living things. I commend these to you. It's important these days to think about biology as an information science. It's experimental, it's observational, but it's about information. Some of the remarkable discoveries listed here are in my lifetime. The first was the recognition, to everyone's surprise, that the molecule of heredity was DNA. It seemed logical that much more complicated, varied molecules of proteins would be better candidates. Classic experiment using pneumococcal bacteria proved that it had to be DNA. And then, as we saw the pictures earlier from Dr. Alberts, came the Watson and Crick, Wilkins and Franklin model of a double helix. The realization, when we could actually spread the chromosomes and see them individually, that we had 46 total chromosomes in our cells instead of 48. The triplet code for proteins. And very importantly, the concept of unity in diversity across all the diverse things, plants, animals, microbes, people, that very many biological processes, very many conserved genes drive the nature of the cells and the organisms across this tremendously diverse kingdom of living things. And more recently, the ideas of systems biology, synthetic biology, are playing in our field. These are the pioneers I just named. I hope you all know who Rosalind Franklin was, as well as the guys. The central dogma of molecular biology took hold in the 1960s and was a driving force to capture the flow of information from DNA to RNA to protein. It seemed a compelling and sufficient explanation for the knowledge of the time. That the genetic blueprint is in the DNA sequence, that's transcribed into messengers that are RNA molecules, and then translated into proteins which carry out most of the functions of the cell: enzymes, hormones, oxygen-carrying hemoglobin, immune responses, molecular motors like Dr. Alberts talked about, and structural features of cells and tissues. But now we realize we've had wonderful surprises as we learn about the complexity of living things. First, information can flow from RNA to DNA, mediated by the enzyme reverse transcriptase. It's actually quite a broad phenomenon, especially in viral functions. Genes can be turned on or turned off by methylation or demethylation of cytosine, the C in the DNA code, or by modifications of specific chromosomal proteins, the histones. Gene expression is regulated by a host of other molecules, protein transcription factors, RNA binding proteins, non-coding RNAs, microRNAs, link RNAs, and transposons and line 1 elements, so-called jumping genes. During the Human Genome Project, there was a lot of speculation, estimation of how many protein coding genes there were embedded in the DNA sequence. And the estimate was 50,000, 100,000, even more. It was known by this time that other organisms, worms, fruit flies, Arabidopsis plants, had 20, 30,000 genes. When the results were published in 2001, the best estimate was 35,000 protein coding genes. But more and more redundancies were recognized of very similar sequences that ended up being coding for the same protein, and the number kept slipping. Now the official number is 20,000, 20,300. This is just 1.2% of the genome sequence. So it's very important to ask, what does all that other 98% plus do? And that's where some of the most exciting developments about gene regulation and genome organization are emerging. One of the things most dramatic is that mammals especially, but other organisms as well, have developed a process of alternative splicing, taking the exon-intron structure of genes, let's say the exons coding for RNA and then protein sequence, and the introns mean the intervening sequences within the gene, and making alternative splice transcripts, different products from the same gene. And then if those are translated, there are different protein isoforms, protein variants from the same gene. This is just barely taking hold, and as people need to ask if they're measuring up or down regulation of gene expression or protein expression, how do they account for the fact that they're actually measuring mixtures? And the mixtures do not have all the same effects. In fact, they quite often have opposing effects. This happens to be a major focus of my own research lab. And we demonstrated differential expression of different splice variants in cancers, for example. Next, while the DNA is essentially the same, allowing for the regulatory phenomena in every cell of the body, proteins are dynamic. That means that the protein level can vary enormously in response to external and internal stressors, and the proteins undergo numerous covalent modifications and specific cleavage, proteolysis. The result is dozens or even hundreds of related structural variants with highly varied functions from one original gene-coded protein. More complexity. The DNA packaged into chromosomes in the nucleus of the cell is not just a big blob or a sphere. It's highly differentiated. Now we talk about the topography of the nucleus, and it affects which genes are active and which genes interact and may undergo translocations between their chromosomes or fusions, as I'll show you an example later in prostate cancer. This has long been uncharted landscape, but it's now feasible to study very clever methods using chromosome chemical crosslinks. So you can convert what was previously just out of reach through fancy microscopes into sequences that can be readily analyzed and map the parts of different chromosomes that are brought into close physical proximity and functional relationship. It's a big item, and there's a lot of signal to noise to be sorted out in analyzing these data. So this is a very nice chart. I'm not sure it projects clearly enough for you. From the 10th anniversary of the Genome Project, let's say of the report of the Genome Project's human sequence. This is in Nature, February 10, 2011. And the highlights go all the way back to Mendel and the experiments on peas in 1865, and then Watson and Crick, and the triplet code, and then the GenBank for data. Very important to have data promptly put into public domain. and then all the different genomes of very diverse species that have been studied. In fact, I remember very well in the 1980s, there was a big discussion that we should study the genomes of other species. Why just humans? But if it's going to be called the Human Genome Project, maybe that would exclude other species. Not so, we said. After all, to understand any one species, it's important to study many and learn from the comparative analysis of genomics, and that's been extremely productive. And you can see all these Science and Nature covers. The summary is here. Methods, data banks, Genome Project launched officially in 1990 under James Watson, and then led by Francis Collins. And then more and more sort of an explosion of results since that time. Now, all of this has to be integrated, and the first level of integration beyond the molecular is the cell. And of course, we just heard from Bruce Alberts, The Molecular Biology of the Cell, 5th edition. You saw him in Antarctica highlighting the release of this 5th edition. So, the anchor for all of us is this weekend, 11 years ago, when Nature and Science published full issues about the human genome sequence and many articles each about the interpretation and application of that information. Do any of you have these issues? They're collector's items. It's a good thing for your library, if not your personal library. And these are the people who were involved: Francis Collins, Craig Venter on the 2 Genome Project teams, Ari Patrinos, much forgotten fellow from the Department of Energy, who brokered the compromise that led to the joint release of all those data, and then the guys who ran the technology, Lee Hood, and the Big Data Generating Center, Eric Lander at Broad Institute, MIT. Lander had a very nice paper in Nature, and there's a video up from this, about the contributions scientifically of the Human Genome Project in the 10 years since the release of those reports. And there's been tremendous impact in genome sequences, in functional elements, in understanding the nature of many, many inherited diseases, and in characterizing the genomics of cancers. The driver for this is technology. Much of this would not have been feasible without amazing advances in genome sequencing. And we're going to hear a lot about that in the next talk. Here's Business Magazine telling us that the genome is going to be feasible to assay for a reasonable price. The really exciting thing of this slide is how the price has gone down. You're probably familiar with Moore's Law from electronics and information technology, which is the doubling of information every 18 months for the same price. And here's what's happened, something similar with genomics until in the last 3 to 4 years, the price has plummeted as new technologies with massively parallel analysis have become quite practical. Looking ahead 10 years, the notion is to build this foundation of knowledge about genome function, genome biology, make diagnostics routine in human medicine as well as animal medicine and plants, and define the genetic components of disease, including common diseases with multiple causation, characterize cancer genomes and genomes for other diseases, introduce practical systems for clinical genomic informatics to be used with the medical record, determine the role of human microbiome, health and disease, as we just heard. So this is my favorite cartoon. What can you see here? This appeared in the Financial Times of London 5 days after those 2 covers of Newsweek, of Nature and Science magazine. You know, the release of the nearly complete human genome. And what are they showing 5 days later? Where is DNA? Can you find DNA? It's off stage in the shadows. Check that off. Genome more or less done. What's next? This is the way the public and the media look at science. And in the center stage is protein, the next holy grail, racing to decipher the human protein set. And that's what we're about, as well as all this tremendous further work on genomics, because we need both. And proteins, as you heard, is an area in which I have invested decades. And here's a bigger way to think about it even, which is multiple layers, starting from genomics up through the transcriptome, gene expression, the epigenome, microRNAs, proteins, metabolites, cells, tissues, organs, the whole phenome or phenotype of the individual. Also physiological information, clinical information. You can start from the environment, the effects of behaviors and chemical exposures and infections, social networks, all that on these phenomena. Or you can start from the systems biology and systems medicine of measuring these and inferring and then demonstrating their effect on phenotypes. This is the future of utilizing this information, especially medically. And we have to realize that when we measure things, we collect data, but we have to take the data into well-organized formats we call information, and then apply it in ways that test important questions. Remember, every speaker practically has talked about the importance of asking the right questions, what Ishmael called the fecundity of good questions, and that's knowledge. And to manage all that knowledge, we have to have people who are skilled to do it. The public is interested in all this. The public interest is very high, and maybe that's why some of you are here. It's not new, however. Almost 50 years ago, the US president took the helicopter from the White House in Washington, D.C. out to the National Institutes of Health and landed in front of the Clinical Research Center, said he admired the biomedical research enterprise at NIH and around the country, but he reminded us that the nation is impatiently waiting for results. They want the research translated into better medicines and better health, and of course, so do we. That's a popular topic with our current president as well. Here's a very interesting thing by colleagues of mine at Vanderbilt University. This is the number of facts that we can manage in making analyses and decisions, especially clinical. And the cognitive psychologists say we can pretty much handle about 5 different kinds of information, preferably 3. But what's happening now is we're having an enormous amount, we have an avalanche of information. So having organized ways of dealing with information that's clinically relevant or potentially clinically relevant is going to be a challenge for how we organize and practice medical care. That's why I emphasize at this juncture that for you young people, among the wonderful fields you can consider, one of them is bioinformatics. All these advances generate a flow of data that will put a premium on those who can organize, mine, model, and utilize complex data sets. It's a new discipline. Another view of this is that we may be overly focused on the genes, because genes do not operate in isolation. We must understand the relationship between genes and gene actions and all these other factors that were in my big genome to phenome slide. This means the fields of public health sciences, statistical and epidemiological methods, nutrition, which we just heard about, and metabolism, lifestyle behaviors, cigarette smoking, alcohol drinking, exercise, physical inactivity, all kinds of exposures to microbes, chemicals, physical agents. And from those, the meaningful use of tests and medicines in disease, and the policies, bioethics, and services of utilizing genomic and other molecular medical information. To do research on those questions requires populations, so having large epidemiological cohorts, of which many already are available, and doing the genomics, or having the genomic studies and doing the studies linking with other data about these other exposures is a valid and important strategy. There are collaborations on a large scale involving big biobanks. Here are a few examples, and there's going to be more and more opportunity in this area. Now, molecular studies and ecogenetics, genetic environmental interactions, are important in infectious diseases. It is now routine to use genomics to characterize infections in people and animals, plants too, I believe. It's practical to probe for which antibiotics work and don't. It's crucial to surveillance, as with SARS and the influenza, TB and other cases. And finally, there's a lot of information about host pathogen interactions in susceptibility and resistance to HIV, malaria, and other infections. On the chemical side, we know we have an organized approach to hazard identification, characterization of risk, and risk reduction. And the key elements here are variation in susceptibility and the need to carefully measure exposures. Let me turn now to common diseases. This is where the big action is in medicine and in public interest. And this is a cover from Science magazine 10 years ago, almost to the day, April 26. A whole class of studies has been dominating the relevant medical literature called Genome-Wide association studies, using SNPs, probes for specific alternative base pairs in the genome, and measuring in present assays up to 1 million of these at a time, and trying to find the ones that are statistically significantly associated with interesting traits, like the ones shown here, or diseases, and many, many others. There are some limitations of this approach. First is, since only 1% of the genome codes for proteins, that very few of these SNPs that are associated with diseases are in protein coding sequences where we can predict what might be the effects and model them with bioinformatics. Secondly, there are big assumptions about saying just because a SNP is somewhere near a particular gene means that it has something to do with that gene and maybe not others. We have very limited collective attributable risk, even when we have 40 associated SNPs for type 1 diabetes, or 180 SNPs for height, stature, still accounts for 10% or less of the variation in risk for that trait or disease. So we need to think how to capture more of the known heritability of these conditions, and one of the biggest is the gene times environment interaction, also gene times gene interaction, and this will be an area of increasing importance. We also have to capture these alternative splice transcripts I mentioned, and of course we will have wholly new data. We already are organized to measure the results from deep sequencing instead of microarray hybridization. Here's an example: obesity. It's one of the really growing worldwide epidemics and predisposes to diabetes, to cardiovascular disease, and one you might not have thought about so much, which is non-alcoholic fatty liver disease. A young woman on our faculty, MD, PhD, MPH, hepatologist, doing beautiful genomic and metabolic work on this condition of NAFLD, non-alcoholic fatty liver disease. So obesity plus tobacco smoking accounts for 1/3 of all the deaths in the United States. In the different states around the country, you can see there were hardly any states that had more than 19% overweight or obese 10 years ago, and now, with one exception, every state has over 20%, and most have over 25 or even 30% overweight and obese. This is a rampant epidemic. And it leads to many, many organ system complications affecting practically every part of the body. Around the world, it's now approaching 1.5 billion people. Just as we heard, there are lots of malnourished, undernourished people, but there's going to be even more overweight and obese. So this is a very interesting condition about fatty liver disease. It's progressive. It has different stages. Each stage is accompanied with different gene predispositions. There's a connection, but not a full correlation, with metabolic syndrome and with cardiovascular disease. So there are many points of intersection for gene, nutrition, environment interactions. And the heritability is about 27% for this liver disease, 40 to 60% for obesity. For body mass index, or overweight, obesity, 5 years ago there was only one genomic marker associated. And then a second one, which is a well-known obesity factor. And look at this, been an explosion, and there's further since then. And these are aligned with very interesting, broad array of biological processes and phenomena which show you the impact of BMI and obesity on so many organ systems. There's a whole connection through appetite regulation, shown here with MC4R and leptin and so forth. But when we turn to the liver disease, so far, there's only these 5. One way back here, and plus 4 more just reported in the past year. There will be more, but these are already very exciting because they're not just statistical associations. These are in or involving genes central to triglyceride, glucose, other lipid metabolism, and this means you can study and investigate with hypothesis testing what might be going on related to these variants in different patients. Also, you can tie together imaging of the liver and really have a comprehensive view of this class of disease. Second example is cancers. This is a very big problem in the United States and increasingly all around the world. A lot of deaths, a lot of new cases. The most common ones, we don't have very successful treatments. The ones we do have very good treatments are rather rare cancers. So here's work from my colleague Arul Chanayan. It's what I call the emergent properties of databases. So bioinformatics is at the center, and all these other approaches are built on it. This is Arul Chanayan, his and our student from our bioinformatics PhD program, Dan Rhodes, built something called Oncomine, all the then available or obtainable data sets on gene expression related to cancers. And they built into this resource called Oncomine a whole host of analytical tools, informatic tools, statistical tools, and they were able to show for this particular event here that when they asked not about prostate cancer patients versus normals, but prostate cancer patients' variation, how much variation was there within the patient population for various genes and their transcripts, this particular gene, ERG, stood out that there were some patients with extremely high expression. So they investigated that, and they found out it was due to a fusion of this gene with another one, which is a sort of ordinary membrane serine protease, but very importantly is under control of androgen, male hormone, which is what drives prostate cancers. And therefore, when they're fused together, androgen hormone drives this gene, which is a transcription factor, part of the ETS family, and makes cancer cells proliferate and grow. And this is the work Scott Tomlin's published in Science in 2005. Well, now, this work has been greatly extended. It's what we now call personalized oncology, personalized medicine for cancer patients. 50 to 70% of prostate cancers have these various ETS family fusions. There's another kind of gene which is in 10 to 15% of patients. And there are these which are in only 1 to 2%. Well, 1 to 2% of 220,000 men per year in the United States is a few thousand each. And what's important about this list is we already have drugs that treat these mechanisms. It's not a matter of finding a new target and hoping that in 15 years a pharmaceutical company might eventually have a successful drug to treat it. This is just the opposite. Here we have the drugs. We are looking for the patients that have the right mechanism to use the drug for them. And this is working very well indeed. And this gives you an idea how many more of these are available now. This is just for prostate cancer. It's an elaboration of what I just showed you. And we have similar lists building for lung cancer, breast cancer, thyroid cancer, you name it. The idea here is to do these studies of the tumor biopsy, genomic analysis, figure out which drugs might work, who needs the treatment, and even what the dose should be. We call this the Michigan Oncology Sequencing Center. It's a CLIA lab. It's going to be service for a lot of patients, and it has other extensive information about point mutations, insertions, deletions, copy numbers, structural rearrangements, if you're interested in the detail. This whole slide set is up in the web for all of you. Another point about cancer patients is, besides making an early diagnosis, especially with breast cancers and prostate cancers, we would like to know which ones are relatively benign and which ones are potentially lethal. So we've built a strategy using metabolomics as well as transcriptomics, and this is information published in 2009 in Nature. And the red over here is for metastatic lesions versus the gold, which is for localized, and the blue for benign. And a little easier to see here, there's huge overexpression of this particular compound called sarcosine, which came out of, again, bioinformatic analysis, saying which pathways were upregulated in association with metastatic prostate cancer. And there was amino acid metabolism, methylation metabolism, and we thought, aha, maybe there's a connection of methylated amino acids, which is what sarcosine is, methylglycine. And this has been turned into a diagnostic test, as has the fusion gene for diagnosis. I want to say just a few words coming down the home stretch about proteomics more broadly. This is a major report on the human plasma proteome, which we put out in 2005 from the Human Proteome Organization, international collaborative effort. And now we've launched a global effort called the Human Proteome Project, like the Human Genome Project, with extensive work already available on antibody-measured proteins and on mass spectrometry measured proteins, databases, and we're looking chromosome by chromosome, so there's 18 now of the 24 chromosomes have already been claimed by competent groups around the world, and there's a companion approach looking at different organs and diseases for proteomic information. And the next big meeting is in Beijing in 2 weeks, and then in September in Boston. And there's tremendous energy into this and national funding from many countries, and I happen to be the chair of this project. It's quite exciting. This shows the biology and disease-driven teams by organ system and disease categories. This is the chromosome team alignment around the world. This is the data pipeline, and this is how we see bringing all this kind of information together in a way it's never been done before, especially from antibody approaches and mass spectrometry approaches and looking at diseases. We happen to be a team for chromosome 17 with a focus on breast cancer because ERB2 or HER2/neu is on chromosome 17, BRCA1 is on chromosome 17, TP53 is on chromosome 17, and we've already demonstrated. If you ask what are the top 20 genes associated with HER2/neu positive breast cancer for gene expression, 13 of those are on chromosome 17, and all 13 of those are co-regulated with ERB2 in this analysis. Finally, there's a report just released from our National Academy of Sciences 3 weeks ago, which I happen to chair, on translational omics. Leaving aside the reasons for this report, it's very timely and there's a tremendous amount of effort about trying to implement the recommendations about best practices for discovery and translation of an omics-based test from earliest development all the way through to analytical validation, clinical utility, and clinical use. And if you're interested in that domain, you should take a look at this report. It's available at the NAS, National Academy of Sciences.edu website, and there's a very valuable framework for science and regulatory approaches and institutional journal funder and practice community responsibilities. So, 12 years ago, Francis Collins said, mapping the human genetic terrain may rank with the great expeditions, Lewis and Clark, Edmund Hillary, and the Apollo program. We think that's true. And it's up to us to make it good by doing more than just the genomes, to understand this dynamic proteome compartment, to incorporate behavioral, environmental, and healthcare variables, and to integrate those variables through proteins. Finally, I say to all of you, especially young people, an inspirational figure in my career a long time ago had this plaque at a time when many people were asking for role models. he said, "Do not ask what path you should follow. Go instead where there is no path and leave a trail." I think that's what Bruce Alberts talked about also. Thank you all very much.
Thank you so much, Dr. Ullman. We have questions, please, for Dr. Ullman. I may take the advantage of being the chair and ask you a question. So extremely exciting, of course. One of the things, as you know, it's kind of rare that a disease will be caused by a single polymorphism or single gene. It's rather the combination of polymorphism genes that can either exacerbate or suppress the effect of that gene. So other than things like WebQTL and some software that has been written for a systems approach to genomics and looking at the overall context of the host, what is going on in terms of advances in getting more tools for this analysis of complex traits?
Yes. Well, this is a very important big question. The question whether multiple genes are involved applies not just to complex traits with multifactorial inheritance, some of it genetic and some of it other factors, as I emphasized, but even to single gene diseases. For example, my colleagues, led by Leroy Hood at the Institute for Systems Biology, are looking at Huntington disease, which is a classic single gene autosomal dominant condition affecting the nervous system. And it's clear that there's a lot of variation among people and families with this particular mutant gene, and it's certain that other genes play on the whole process. So they are looking with genome methods, with transcript methods, with proteins and metabolites to see if they can characterize both the downstream effects of the Huntington primary genetic lesion and the influence of many other genes and pathways.
Yes.
Now, if you're just looking at lots of genes, it's a big number.
That's right.
But if you can organize it around pathways and networks, that gives you functional meaning and an opportunity really to figure out the answers to this question of how is it mediated, how is it modified when you're looking at multiple genes and multiple gene-environment interactions.
Thank you very much. Any other question? Yes, please.
Go ahead.
Is there a microphone?
You need to use a
please?
microphone. There's somebody at the microphone up here.
I've got a very quick question, Gilbert.
Yes.
I'm interested in where the microbes in the body fit within your paradigm of disease linking to genetic information, and if you think the microbiome is important.
Absolutely. So I had them in at least two different places. One where I put about the surprises and complexity that the microbiome needs to be measured and taken into account. And the other is about microbial and chemical and physical agents that play on the genetic inheritance. Now, one of the fascinating things about this is, as was said earlier, that the microbes are detected by their genomes. We are still teaching lots of places that the pulmonary tree, for example, is sterile. It only gets infected when you have an infection. But it turns out that the whole surface of every surface on our body, but including the surfaces through the trachea and down to the bronchi and into the lungs, are completely covered with microbes. It's just that we don't have methods to culture them or grow them, but with genomic analysis we can show that they are there and that the composition of species changes dramatically under certain conditions, like patients with chronic obstructive lung disease, where Pseudomonas species appear in big percentages which were not readily detected previously. So this is a huge new variable, and a lot of labs are working on it. Our bioinformatics team at our center is working with pulmonary, skin, gastrointestinal, immunological groups at our university, and with ecological colleagues in the rest of the university outside the medical area, figuring out better ways to analyze the genomics and extrapolate to the likely functional effects. And of course, we think it affects nutrition, we think it affects drug absorption and drug action. It's a big story.
Okay, I have a question about the PSA. You explained now that proteomics is under development and they have a lot of results. What is in the point of view of the analyst in the laboratory? Being a professor of clinical biochemistry and immunology, my name is Tafa Nasr anyway, I look forward for markers earlier which we can detect the cancer in case of prostate more efficient than the tumor markers. And what is your opinion about the gene therapy in such cases? Thank you.
That was a very complicated question. I'm not sure I got all of it. I think you were asking about protein biomarkers, and at the end about gene therapy. Is that right? Okay. Well, we don't have any gene therapies for these conditions at present. In the United States, there was a big problem that arose from a premature use of gene therapy in a young man who had liver metabolic trait, and he died, and it set back the regulation and approval of gene therapy studies in our country. But there will be work coming along as we learn more and find ways to deliver it. There are some actually very exciting ways of modifying adenovirus vectors to make them safer for delivery so that they don't actually cause disease themselves. As far as protein biomarkers, it's been a challenge. There's not very many that are in widespread use. There's one that's sort of curious involving 5 quite abundant proteins in the plasma circulation, which is used for ovarian cancer patients to determine whether the surgeon should be an ovarian specialist or a general surgeon. May not seem like a very big issue in general, but to the woman needing the surgery, it's a big deal. And since only 15% of those big pelvic masses are ovarian malignant tumors, it's important to know what is the cause. Many other markers are under evaluation. There's a big early detection of cancer proteomics effort supported by our National Cancer Institute and by other disease institutes. So this is a work in progress, but our methods are getting better. The spliced transcripts, spliced proteome variants, I think, are going to be interesting in this regard. And the output from this Human Proteome Project will surely accelerate that work.
We have time for 2 more questions, so please go ahead on the other side.
Yes. I would like to ask if behavioral activities were related to the genetics. Like there are data on the genome that control the behaviors we exhibit?
Whether genes control our behaviors or whether we change our behaviors if we know our genetic information?
No, do genes control our behavior?
Yes. Okay. We are certain that genes control our behavior. Influence our behavior might be a much more appropriate word because there's a lot of other variables and there's some degree of free will. We have data, for example, the behavior of smoking cigarettes is influenced by the addictability of the habit, so that receptors involving dopamine have variants that are genetically determined, and that's associated with greater or lesser likelihood of becoming addicted and becoming a regular smoker, for example. And then there are genes predisposing to many behavioral and neurological conditions. But there's another side of this, which is with all the direct-to-consumer advertising and people sending their samples to get genotyped, does having genomic information change people's use of healthy behaviors, may help motivate people to do what would be in their own self-interest? This is a pretty interesting question. There's a whole symposium in the Annual Review of Public Health 2010, which that was the wrap-up paper. And the conclusion was that it's likely that it would be used in that way by people, but we have very limited evidence, and the article is mostly about how you would design the studies to ascertain whether these kinds of consumer-based genetic testing would really be beneficial to people. It falls under the category of what we call comparative effectiveness research. Finally, there are pharmacogenetic approaches where we test for the response or failure to respond to certain drugs that are used for depression or schizophrenias or other conditions. And there's definite evidence that we can show which patients genetically are more likely to respond well or not respond well. And this is likely to be very useful. It's already practical at a small scale because these drugs take weeks sometimes to make a judgment whether you think they're helping the patient or not. So to know which drug you should try when there are multiple choices available can be very useful. Okay.
Thank you. I think we should move on. Do you have a quick burning question?
Okay.
Okay, go ahead, since you're standing there.
I'm asking about aging, why we are getting old. Is it true we are still young because our DNA is still young and acting actively because it's still normal or young, and we are getting older because DNA is decrease in activity? Is it
That's a little too hard of a question. Basically, the DNA is neither young nor old. The DNA does accumulate mutations, and maybe that contributes to disease risks. But there's been a project for a long time in the U.S. supported by the MacArthur Foundation called Successful Aging, to try to understand what goes on in people who get to age 80, 90 plus in very good shape, as well as those who develop specific illnesses. The general notion is that if we didn't develop specific diseases, most of us probably would last a very long time.
okay? Thanks.
Good luck.
Thank you so much. Thank you for the question. Shut up.
Thank you, Chairperson. Thank you all. I'm here for the 6th time. I have learned a lot from this meeting. I understand more what is the right thing done by the right people in the right place at the right moment. This is the BioVision chaired by Dr. Ismail Sarajandin, attended by us all in Alexandria, and at this important moment in Egyptian history. I will be here only for 6 hours, even though I have much longer time in the air. I have brought the best regards and the deep respect, firm support, and the full acknowledgement to Dr. Ismail Sarajandin for his great contribution to the global sciences. That's from the bottom of my heart, also from so many friends and colleagues in the international scientific community. I would say the Egyptian people, especially young people, have all the reason to be proud of him. I will talk about genomics. No talk about genomics could be started without talking about the Human Genome Project, as many speakers have done today. I would say it's the first time for us to look into life at the whole genome level and at the base resolution. The biggest and most significant impact of the Human Genome Project on life science would be and omics. Within the next 25 years, omics will be the biggest, if not the only game in town. I would not sing other praises for the Human Genome Project. I would say it would be regarded as a mission impossible. I would quote from one of our colleagues, either we will never be able to know the complete genome sequence of our human being, or we have to wait for 3 centuries. It's not right. We have done it. More importantly, we have done it together. Of course, still something we would feel sorry. It's too slow, it's too expensive. More than 3,000 people in 16 centers in 6 countries, and then took 13 years at the cost of $3 billion just to complete a single individual's genome. Now, the situation is quite different. Even in my own center, BGI, we have more than 100 high sequencers, and we are able to do the same job of 100 human genomes. Until the end of last year, we have already, together with our colleagues, delivered more than 600,000 gigabases. It's equivalent to 200,000 human genome size. This is an opportunity. Sequencing is a tool for us all to understand the essentials to life. I fully agree with the editorial by Nature Biotechnology that the genome sequencing has provided a starting point of everything, and ultimately genome sequencing is the digital foundation onto which all the other studies can be placed. We can understand life in all different ways. To me, I would like to share my beliefs with you again. First, life is in sequence, off sequence. Then life is digital. If we put these two languages together, we can see obviously that life is digital in its nature. Based on these two beliefs, we have the two strong platforms. One is sequencer, the other one is a computer. Sequencers and computers, only hardwares, we do have powerful supercomputers, but more importantly, that's the bioinformatics, which is the core competitive or technology of genomics. We have written many softwares. Some are not so good, some are equally good as yours, but we do have a few which are evaluated as the best of the best. More important than these two beliefs are not the two spirits or souls. The first is the AGP spirit, as it is called, that's owned by all, done by all, and shared by all. Then we do have the culture. Whenever we think of sequencing something, we think of collaboration. We always say genomics cannot be done alone. Genomics is international. Let's do genomics together. I would give you a brief story about the 3 stages in my institute's history. That's true. BJ was born for a small part of the Human Genome Project. We joined the Human Genome Project in September of 1999. This was really a big job, or a dirty job, at least. We counted, not only counted, but used up 50 million tips with this kind of machine. BJ was known by working on the rice genome. We published as the first time by mainland Chinese in Science. This paper has been quoted more than 1,000 times, and it has been evaluated as one of the landmark papers which should be read by all plant biologists. We can see from this figure how the release of rice genome sequence data changed the situation of the researches on 2 important crops, rice and wheat. It is even more significant in those developing countries. There might be other interpretations to these figures, but our colleagues in the International Rice Research Institute told us the reason is no studies can really be done without the genome sequence of that organism. Of course, the reason for us to have completed the right genome sequence by the strategy of whole genome shotgun is we developed these important softwares, of course, which have been published and are freely available. The real job we have done together with our collaborators is with the newer generation sequencers. If our contribution to the human genome sequence is so small, at the end of 2008, we published the whole genome sequence of an Asian individual. Scientifically, it tells us that human diversity is much higher than we expected. Technically, this job told us that the technologies available now are extremely powerful. The real breakthrough, as called by our international colleagues, is the job on the giant panda. Without the help of genetic map or physical map or any knowledge of the repeats, we successfully reported the giant panda genome. That's the first de novo assembly of a large mammalian genome achieved only using the next or novel generation sequencers. These three stages in BGI's history just correspond to the three breakthroughs in sequencing technology, from hand sequencing to automatic sequencing, from slab automatic sequencer to capillary sequencers. And then now we have the massively parallel, and this revolution is still continued. What are we doing now? First, sequencing for the reference genome, as we have done for the rise for panda genomes. We have published the genome sequence of 2 monkeys in Natural Biotechnology, and then in the same journal, several cell lines of CHO, which are important for genetically engineered products. We also published, together with our colleagues in the States, one of the naked longevity mole rat, as well as 2 species of ants. We have released the sequence data for the polar bear in the North Pole, penguin in the South Pole, as well as the Tibetan antelope on the roof of the world. Based on these results, we have initiated the international collaboration on sequencing 10,000 vertebrate genomes. It's about a quarter of all the known vertebrates in this world. On plants, we published the potato genome in Nature, and the cucumber genome in Nature Genetics, and also the Chinese cabbage, as well as Indian pigeon pear. Before the end of last year, we have sequenced the reference genome, that's an individual of that organism, more than 500 species. And then we are going on. This is the job in collaboration with our European colleagues. Now we know that in every of us, there would be more than 170 species of bacteria. In all, at least we have identified more than 3 million genes operating frames. It is about 150 times that of the estimated number of our nuclear genome. That's the reason we have initiated the 10,000 microbial genome project to sequence most, if not all, of the identified cultured microorganisms. And then at the same time, we initiated that Earth Microbiome project to sequence the mega genomic samples, at least 1 million, collected from all over the world based on the Google map. Until now, we have sequenced more than 3,500 microbial genomes, as well as about 2,000 of metagenomic samples. That's our ambition. That's the dream shared by many of us, to sequence every living thing on Earth, every single species you can think of it being sequenced, and not only just one, but many samples to rebuild the tree of life. We can say something similar. Nothing in biology makes sense except in the light of genome sequences. The second, sequencing for genome diversity or variations. This job began with the Human HappyMap project. Now we have just published the first paper in Nature from the International 1,000 Genomes Project. We have identified at least 37 million SNPs, still with rather or relative high frequency, which is much more than we expected ever before. And then we have also sequenced the identified descendants of Genghis Khan. Perhaps you have read this story, the genetic legends of the Mongols. We concluded that about 1 out of 200 of the males carry the Y chromosome or the Y chromosome haplotype, of gene hindsight. We also published this paper in Nature Biotechnology named Human Pangenome. It indicates there are many sequences in our genomes which are population-specific. We also help, by means of de novo assembly, to have compared the genome diversity of several populations. It is true, in addition to SNPs, other structural variations are also more frequent than expected. We have compared all collected DNA sequence with their transcribers, and we found that RNA editing, the difference between DNA and RNA would be higher than we expected too. We have done something on evolution. For example, we have found at least one gene which has a significant frequency difference between Tibetan Chinese and Han Chinese, which could partially interpret why Tibetan Chinese have been able to adapt them so well on the high altitude. That's the reason we are going to sequence at least 250 small pedigrees in collaboration with our colleagues in the Netherlands. Now we will wait for the final results. 10K British genomes, 50,000 Danish genomes. This idea has been accepted by our European colleagues. And how many Chinese individuals should be sequenced in order to know one of the biggest population in the world? The estimated number is 1 million. I don't know how many years it would take us to complete this job. Definitely, I have all the hope on the young generations. Then the third, sequencing for defined phenotypes. This job began with our sequencing project on the genetic variation map on chicken. We sequenced one strain for meat, the other strain for eggs, and then another for medicine. Several years ago, we published only one strain of silkworm's genome. Now, in a sense, we have published at least 50 genomes of both wild and the domesticated silkworm strains. And then we have sequenced more than 100 and analyzed. We have published 6 elite strains of corn, as well as more than 30 wild and cultivated soybean genomes in Nature Genetics. Now we are going to sequence more than 1,000 collected strains of soybean together with our American colleagues, as well as 10,000 ice cultivars in collaboration with IRRI. We have published at least 50 rice strains in Nature Biotechnology with very interesting analysis. In all, more than 25,000 individuals or strains or cultivars of animals or plants have been sequenced. Sequencing for applied genomics. Just to give you 2 examples. First, agricultural breeding. We know that the melon or foxtail melon might be the first domesticated crop in China by Chinese. It has a shorter life cycle, at least in the greenhouse for experiments, only 2 to 8 weeks. It can save a part of energy. You can see sorghum is taller than maize, and then foxtail millet is so short. We have sequenced more than 500 hybrid populations, and then we have identified numerous markers, and we have put them together into 9 linkage group, anchoring more than 80% of the sequences. This job, because of this relatively small genome, was finished in about 2 months. Then we sequenced about 100 strains of different origins or different regions of millet, and then we mapped at least 7 important loci which might controlling important traits. Then, with the help of the markers, so-called marker-assisted selection, and then we began our breeding of mallet. It is not so bad. At least for one of the strains, we have got the yield of 12 tons per hectare, and then it can be drought tolerance. And then a few examples for medical application. We published this paper in Nature Genetics that exomic or coding sequencing would be a powerful tool for monogenic disease. We published a series of papers, and we initiated 1,000 Mendelian disorder project in 8 or 9 years. We are going to sequence many, if not most, of the monogenic diseases, especially their family or pedigrees. We have initiated, and it's going very well, to sequence 10,000 autism genomes. We have done something as the first step to explore the cancer genomics. We already have published 2 papers in Nature Genetics. One revealed that the ubiquitin-mediated proteolysis pathway might be involved in clear cell renal cell carcinoma. And the other is about the chromatin remodeling might also be involved in the blood cancer. Now we're able to sequence the genome of a single cell to reveal the high heterogeneity of a cancer sample. We already have published 2 papers in Cell in the same issue in collaboration with many labs. This paper will be published next week. It is the first large-scale investigation to reveal the possible relationship between the HBV infectious and liver cancer. As a part of the International Consortium of Cancer Genome Project, we have already published a paper in Nature. Of course, our contribution is only on gastric cancer. In all, until now, more than 40,000 human samples, both normal and diseased, have been sequenced. Perhaps you have seen this paper in New England Journal of Medicine. That's about the pathogenic E. coli last May in Germany. We also have published this paper in Nature. We got more than 80% of that diploid genome sequence from a few hairs preserved in a museum in Denmark. That's also a cover story in a sense about one of our jobs. It would change our knowledge about the possible evolution and migration of Asian people. The material is still old hair. Now, we have done another job for early stage and non-invasive prenatal genetic tests. We have done more than 20,000 cases. It seems it works very well. It is totally non-invasive. Only 3 or 5 milliliters of blood drawn from the mother. Scientifically, we have published a series of papers. At least this one, which is very important, that softwares or bioinformatics is very important for the high throughput and the deep sequencing of the maternal blood for the genetic test of the baby. Perhaps we can say now sequencing are developed in 4 directions: southern sequencing for DNA or genome sequencing, northern sequencing for RNA or transcriptome sequencing, western sequencing to start the protein or promics by sequencing, of course, with the help of antibodies, and eastern sequencing. That's for methylome. We have done the job on the silkworm methylome as well as human beings. This is an important project. We will sequence the genome and the methylome of 5,000 identical twins in collaboration with our colleagues in the UK. We will see what will this project tell us. The 21st century was regarded or is regarded the century of biology. Let's embrace this new century. Now we have the technologies. First, molecular biology technology or gene manipulation technology to use either DNA or RNA, which would bridge molecular and cellular levels of researches. Then the second, we're able to induce embryonic or adult stem cell into differentiated tissue or even organs. We're also able to induce or reprogram the terminated differentiated cell back to something like stem cell we call iPS. Zhou Qi's lab in China has demonstrated that it is true, iPS still keep all the information for a whole individual, and our job published in Cell also indicated lower incidence of DNA sequence variation in human iPS cell lines, which are useful. Now we know it's not done by us. Human kidney created from stem cell, and people could soon grow their own garden after human kidney created from stem cells. This is a synthetic biology, which has already been mentioned in previous speeches. The program of life was written 4 billion years ago. It's time for whom? For us to rewrite the program. Dr. Craig Venter has done the job. The first genome was designed and synthesized by people with their brains as well as a computer. I said just a few months ago that now we're able to design and synthesize a genome similar to Cynthia, about 1 megabase, and in 3 or 5 years, 5 megabase, in 8 or 10 years, we might be able to design and synthesize the first karyotic genome. It's too conservative. We already have announced on April 16 that in 3 or 5 years, we will design and synthesize the first eukaryotic genome in the similar size of a yeast cell. Now you can see we are sequencing. And then synthetic biology is also to make or design the genome sequence. We are at this important area, at a turning point from reading genome to writing genome. Perhaps you have just heard of another animal has been cloned with something new. Many years ago, Polly, the sheep, was born. Now, most, if not all of the mammalians can be cloned. We announced that we cloned Pong Pong, the first handmade cloned transgenic sheep. It is cloned in a simplified way, and then this sheep contains a gene associated with a well-known unsaturated fatty acid. It is supposed to have this unsaturated fatty acid in her milk. This method is rather simple. Anybody is able to learn it. You just cut an egg with a pencil sharpener and then fuse half of the egg with any cell culture without that complicated manipulation. Then with electroporation, you fuse it and then do just as the traditional way of animal cloning. We have cloned at least 500 pigs. The reason to clone pig is that First, pig is closer to human being in many respects. Then second, the biggest advantage of cloning pig is you can get its eggs from slaughtering every day. There is a famous pig called Pig the Strong, which survived 36 days during that terrible earthquake. Now we have cloned 6 of them. I cannot say whether these 6 piglets are that Pig the Strong himself, itself, or their sons or daughters. This is very powerful. If we combine transgenic and cloning, this is the cloned fluorescent pig. You can see the fluorescence is really strong. The major purpose of cloning pig is to create animal model for diseases. We have cloned several different types of model for Alzheimer's diseases, and even more for breast cancers. If we put them together, molecular biology technique, and then stem cell iPS technique, and then man-made cells, as well as the genetic technology, and what will happen? I would not predict what will happen. I would say that the genomics is the first step. It's the foundation for all we are going to do. Genomics in the foundation of life sciences about biotech, but providing knowledge of genes, genomes, and the knowledge about the metabolic and the signaling pathways, as well as the gene regulation network. This is genomics. I would like to quote from James D. Watson again. All biology in the future will start with the knowledge of genomes and proceed hopefully. I would like to quote from another colleague. Just as physics shocked the world in the 20th century, now it is clear that the life sciences will shake up the world in the 21st century. It is now. This is BGI. According to economists, it is now the biggest DNA sequencing lab in the world. And then the capacity of BGI surpasses the combined capacity of all genome sequencing in the United States. Perhaps that's not true. And BGI is called the sequence factory for the whole world by nature. I don't know whether a factory is a good term or not. And BGI is ranked as the 4th most influential research institutes in China, and then the 7th in Asian Pacific area. According to Science, last year, BGI enhanced its reputation as the world's largest sequencing center by doing this and that. Just this year, 2 months ago, again, Science says BGI has emerged as a genomic superpower, even though I don't like the term superpower. That's true. We have, especially several labs in the world, taking the world by storm. Don't worry, it's totally wrong. We don't have the ambitions, we don't have the ability. But it is true. In 2004, BGI's role as argued change in Chinese science and technology. And now, as a report, that the BGI may well create a very different model. Many people ask me, what is the BGI model? I say, BGI model is no model. But there is something we can share. First, to be humble. As we say, there is always another layer in the sky which is even higher than the layer we can see or reach. Then there is a responsibility. As talked by many speakers, we did contribute to the war against the SARS many years ago. We also contributed to the war against the lethal E. coli in Germany. We called again and again for sharing of the data and the material as we did it by ourselves. We at least did more than 1,500 disaster the victim identification during tsunami in Thailand in 2005, as well as the earthquake in 2008 in China. We are still lagging behind. We are still developing, but we should build our confidence. I'm so proud of my young staff. Now there are more than 4,000. They are working very hard. I would not show you how hard they were working. I just show a picture how tired after they were working. Again, the 3 generation leaders of BGI have been introduced. I'm confident of our young staff. Second generation leader, Wang Jun. And the third generation leader, these BGI researchers are smart, confident, and for their age, tremendously experienced by nature. And then these are so young, they are in their 20s. They will be the future of science of the whole world. We are confident of the future of science in China, Egypt, and in developing countries in the whole world. That's the reason we have written texts for kids, textbooks, and we have summer schools every year for the kids at different ages. We know sequencing is still controversial, but we believe that life is in sequence, life is digital. We will keep the spirit of HGP, done by all, shared by all. This is a cover designed by ourselves. We can only eat a little, but we would like to make so much together with our colleagues in the whole world and for all the people. That's the reason we're still discussing how to release the data, how to promote global genomics. Genomics cannot be done alone. Please be reminded, when I say we, of course, not myself, not BGI. It means BGI and all its supporters, collaborators. BGI strategy is to promote global genomics through the vast international collaboration. I always say, to evaluate the collaborative project to see whether it's successful or not, is to see finally if all the participants become friends or not. We have friends all over the world. I'm confident that I will make more new friends this time at this meeting. This is the core.
Dr. Yang, can we
Okay.
wrap up, please? Yeah.
When you drink the sweet water from the well, don't forget who dig it. A drop of water should return with a fountain. We have established our lab in Copenhagen, and we have even now twin centers. And then we have 3-generation friendship with Sanger Institute in UK, and many former teachers now are working for us. I would like to thank all collaborators, supporters. I would like to thank you all again for your attention. Thank you.
Thank you, Dr. Yang, for a very impressive presentation. Any questions from the audience? Yeah, can you go to the microphone, please? We have time, please. We have time for 2 questions. 3 max.
Okay, please.
My question about one of rare types of cancers, that is dendritic cell lymphoma, and it's resistant to the chemotherapy and radiotherapy, and actually one of my dear friends suffers from it. And I would like to know if there's a genetic therapy for it. And thank you.
I would say I'm one of the most conservative participants in the International Cancer Genome Consortium. I do think that the genome or genomics can help us a lot understanding the carcinogenesis, but genomics cannot really solve the problem of cancers. But it would provide the tools for 2 purposes. One is early diagnosis. The other is instruction of using drugs. And we were going to have another meeting, that's the 2nd meeting this year, talk about what roles genomics could play in the studies and the treatment of cancer. Thank you.
Second question on this
Excuse
side,
me,
and then third
I
here.
would like to ask you two questions. In fact, the first one that you haven't mentioned anything about bioethics in manipulating genes. The other question that I have been to China in the IBS, the last one, and I found that you have taken the cotton seeds of Egypt, of the lens fiber, long fiber, and manipulated without our approval to sell it as a Chinese cotton. Thank you.
I
Next
really don't
question,
know the details. First, for the bioethics, we also have IRB. I myself have been involved a lot. Then, for the samples taken from Egypt, that's a very serious issue.
I am
Would you
informing
please
you
just
because-
write to me? I will give you the clear and direct answer, definitely.
Okay, you can look for that
Sure.
because it was a company and it was from the university and they presented that. And when I contest, they said you don't have a genetic bank, you cannot prove anything. That's one thing. The
we're
other thing.
only addressing scientific
Sure.
questions. Please,
Okay,
you can
this
have
is
this
a scientific
conversation.
question. What
private.
about the bioethics in manipulating genes? You haven't mentioned anything, especially in cloning. Thank you.
But for the data, we
next
do
question,
have released
please.
a lot. For the
Please go ahead
cloning
and
now,
ask your
we
question.
only
Tell
have
them.
done the jobs on the pigs. We have never touched any cloning of a human being, or we have never had the intention. Thank you.
Thank you, Dr.
My name is Salah Soliman. I'm professor of pesticide chemistry and toxicology with Alexandria University and advisor to Bibliotheca Alexandrina here. Dr. Yang, I really appreciate your being with us here and all the data you have submitted about your institute. A couple of days ago, actually the 22nd of this month, a report was released by your own institute while you may be in the airplane about the handmade cloning and the discovery in that area. And I really appreciate that you have mentioned this to us here, and I hope that every country, every scientific society or community just putting back in their mind that science and science benefits should be directed to all, especially with the life science, because we all share in this. So thank you very much again for being with us.
Sure. I actually released it on just before I left China. Generally speaking, we always release and share the information with others. There are 2 points for this job. The first, the procedure of cloning an animal has been significantly simplified, and then now the cost is rather low. Then the second, how does a genetic manipulation technology could be combined with animal cloning? But of course, for the developing countries, actually we have had several, training courses. We would like to share the technology with us, because with you all, because animal cloning is not the major products in BGI. Thank you.
Can I ask you one last question since
Sure.
the third person left? Are you taking RNA from some of those patient samples, from different parts like liver cancer, and in addition to sequencing the DNA, sequencing the RNA from the same individuals?
No, yes, for that paper in Nature Genetics about RNA editing, it's very easy because we have a big collection of both DNA and RNA sequences. Then we just compare, but to exclude the possibility of individual variation. Then we give a very clear or defined number, how frequent RNA editing would take place. But generally speaking, to sequence a new organism or species, we do take both, because the knowledge derived from RNA sequencing or transcriptome sequencing would help a lot for the assembly and the later annotation. So generally, if possible, we do both. But for that early stage of prenatal diagnosis and the non-invasive, we only work on DNA. Thank you.
Thank you so much for a wonderful talk. Thank you.
Let's meet again in 2 years. That's 2014 at the BioVision in Alexandria.